Okay, what I'll present to you is more technical approach. I'm not neuroscientist, so I cannot teach you about that. But tractography can be used to study structural brain connectivity in from a point of view of neurology and also neuroscience. So I think it could be useful for you. Okay, you know better than me that the brain has white matter and gray matter, and what is measured with diffusion tractography is mainly the connectivity given by the myelinated axons of the neurons, and we can see the main pathways. Okay, that's very important. We cannot see individual fibers, neural fibers, but what we see is an estimation of the main white matter pathways. We for the analysis we subdivide them in association pathways that connect regions from the same hemisphere, and we separate them in deep white matter and superficial white matter, because deep white matter are the long, well-known bundles, and superficial white matter are the short association bundles that are more difficult to study. And also there are the commissural pathway and the projection pathways, but I will talk you most about association pathways. Okay, so diffusion tractography is based on magnetic resonance imaging, but we base it on a special sequence that measures the motion of water molecules. And and so for it can to have an insight on the motion, three-dimensional motion of the water, you need to acquire several directions. Okay, not one, but several, at least 6, but ideally 40 or 60 directions. So if you have this measure of the water molecule motion inside the brain, you can apply then a model in each voxel, what is called diffusion local model. And the most known model is DTI, or diffusion tensor imaging. And this model use the diffusion information for several directions and create a model that can be thought like an ellipsoid, where it represents the main diffusion direction in each voxel. Okay, please ask me any, feel free to ask me any question. So I have an image that I think is better to understand that. For example, here in a slide, so you can see that the diffusion tensor model can be seen as an ellipsoid, and you see in regions where you have water, or I don't know, in the cortex also, the diffusion is isotropic. So because the motion of the water molecules is the same in all the directions. But when you have white matter, the fibers, the water will move following the direction of the fibers. So this information that is used to reconstruct the main white matter fibers, what is called tractography. And also, there is an important measure that can be calculated from DTI, from the model, and is the fractional anisotropy. So we calculate the tensor in each voxel of the brain, and then from each tensor we can calculate the fractional anisotropy, that is a measure of the anisotropy of the tensor That means that if you have a more elongated tensor, you will have more anisotropy. That is the case, for example, of the corpus callosum, that all the fibers are very well packed and all follow the same direction, so you have a high anisotropy. Okay, so this then fractional anisotropy is a measure based on diffusion. Is an image, 3D image of the brain, and with that, it has been used to estimate the white matter integrity, and a lot of work are based on this measure. So when we calculate the tractography for the whole brain, for all the brain, we use it in all the white matter, and we calculate all the fiber that we can reconstruct with the information we have. And we have this kind of data that are, for example, I think about 1 million fibers. So what we want to do is to study this information and try to study the structure of white matter based on this data, to perform population analysis and to study what is known as the human brain connectome, and is a representation of the connectivity like a graph. There are several methods. I just mentioned you the method that we can apply. Maybe you know some of them, but this is just a description of the several methods that can be used with tractography. The main method is bundle segmentation. That means that you have a model of the bundles, and you can extract these bundles in individuals to perform analysis. Normally, commonly, these analyses are based on fractional anisotropy. That's why it's so important. Why? Because it's different to compare for example, the volume or the shape or the bundles. But fractional anisotropy is a measure of the microstructure of the bundles, so it's more informative than the fibers themselves. Okay, so there are different ways to reconstruct the bundles. You can have manual segmentation automatic segmentation based on ROI atlases, or fully automatic segmentation based on clustering. Also, you can have a fiber clustering method. Clustering method, if you don't know, I just explained that it's a basic, very classical machine learning approach to to extract similar elements from a group of elements based on a similarity measure. So we need here for the fibers to define a similarity measure between all the fibers, between each pair of fibers. So what we use is a distance here that use the point, because the fibers are defined by point, a number of points. I don't know, for example, separated by 1 or 2 millimeters. And from this point, you can estimate the similarity between two fibers. For example, what we use is the maximum Euclidean distance between corresponding points. That means that we calculate all the Euclidean distance between the points, the corresponding points, and we select the maximum. This is a measure of how similar are two fibers, but there are a lot of different measures that you can use. Once you have this measure, you can apply several kind of clustering methods to regroup fibers. What we can obtain with the fiber clustering is groups of fibers. Okay, so these have no anatomical information. If you apply a clustering, you will not find anatomical bundles, but you will have a better representation of the information. So we use this kind of analysis, and so what we normally use are this kind of approach. First, we apply a clustering to reduce the amount of information, because 1 million of fibers is too much information, so we reduce it using a clustering. And the clustering allows us to represent the cluster with few fibers. I have here an image, it's better to show it. So we can have here, for example, I don't know, 100 fibers, but you can see that all the fibers are very similar. This is a cluster that can be found automatically with the algorithm. So we can replace that with only one fiber. This is a simplification, but when we just want to find patterns, it's enough to find the main patterns of connectivity. So this reduction has made available the analysis for a database. Okay, because if you have 1 million fibers and you want to analyze 1 million fiber per, I don't know, 100 subject, is too much information. So the centroids allow us to reduce the information and then apply our other analysis to a database of, for example, 100 subjects. And with this analysis, we can then extract information that is representative of a population of subjects and create atlases of white matter. And also the third step is, once we have atlases, we can extract this information and segment the bundles. Okay, so we have created a first clustering, and what I want to show here is how works a clustering. Clustering use the information, the fibers in this case, needs the calculation of a similarity between the elements. Here the elements are the fibers, and then a clustering normally, not all the clustering, but several clustering use a distance matrix between all the fibers. So you have here the distance between all the fibers. This is a symmetric matrix. And then you can transform this matrix to an affinity graph. You can use a threshold to eliminate the very low, high distances, and then you construct an affinity graph. In this graph, you have connected all the fibers that are very similar, and the edge here is the similarity between the fibers. So there are several clustering methods and other graph methods that can, based it on the graph, can find, for example, the groups of similar fibers. So this example here of the hierarchical clustering that use the graph to create a tree of similarity. And the advantage of this kind of analysis is you can find several, you can cut the tree in different parts depending on what you want to do. If you cut the tree very low in the base, you will find a lot of small clusters, but you can try to find more bigger clusters. So it depends on what you want to do. So this is a classical approach, but we cannot apply this approach to 1 million fibers. It's too much. It's too much. The algorithm will not do that, okay? Because we can apply the clustering, I don't know, to 100,000 fibers, maybe, but not more than that. So the strategy was to reduce the information. It's very simple. I will not detail that, but for example, just using a brain mask to separate left hemisphere, uh, right hemisphere, etc. And also based on the length of the fiber, etc. And several other algorithms, we can extract groups of similar fibers and finally find what I show here is a representation of the tractography based on a group of clusters. Okay, that is the main idea. It's just a representation with less information, but it's very useful for the processing that I will show you. So this was one approach, and we have a very new method that performed the analysis based on a totally different way, but found the same kind of result. It used the point information to regroup the points of the fibers and then extract the bundles. But the advantage of this method is that it's very fast. So, for example, for 1 million fibers, it takes less than 1 minute. Okay, compared with 2 hours for the first method. So here what we try is to have the same quality and have a very fast algorithm. I have here a video of what we found. Here we have the bigger clusters. No, only 200 cluster we have here. We find 3,000. So here are the bigger, but we have a lot more when we apply the clustering. So if you look at the fibers at the cluster, you can see the main white matter bundles. You can see that they are present. So the idea is there are a lot of works based on that, on trying to extract the structural information, but not from only one subject, but from a group of subjects, and then to infer atlases of white matter bundles. I'll show here one approach that we did, but there are a lot of approaches. Here we apply the classical also clustering that I show you, the hierarchical clustering, but it works very well with this kind of data. The only thing here is to find a cluster is not enough to say that it is representative of the population. We had to select only the cluster that had a good representation. It means that these are present in almost all the subject, at least half of the subject. And we found this kind of clusters that represent a pattern of connectivity that is present in almost all the subject. And then we label it these clusters and according to the anatomical description of several bundles. This is a quite old work, so there are a lot of new works that have done the same thing, but this is one example of atlas, white matter atlas. And this atlas, okay, we have the main bundles. Maybe it's not perfect, but it is a representation what we found in this database. And with these bundles, with this atlas, we can segment bundles in new subjects. That is the advantage. And here again, we need another algorithm. The algorithm that we use is very simple. It's not really machine learning because it's more simple than that. But the idea is the main idea of machine learning here is classification. Is the idea is to given a new tractography data set of a new subject, and given the model here, we will try to identify these bundles, the bundles of the model here of the atlas, in the new subject. And this is what we call segmentation. And with that, we can identify on a new database. And this kind of approach can be used for any study, brain study using tractography. Here is only one example with bipolar disorder, but it can be applied to any study, and also combined with fMRI information or clinical data to try to correlate with clinical data, etc. So here, just to explain how to apply this method. Once we have the tractography is calculated in the space of diffusion of each subject. So, and the atlas is in another space here, in the Manai space. The subject is in another space here. So we need to transform, and this is not easy. I don't know if some of you have worked with that, but it's not easy to apply a transformation that will work well. There are several software, but you need at some point to have a manual intervention. It's very time consuming. But once you have the registration here, the algorithm will segment the fibers. Okay, we have several steps and we'll extract the fibers. Once we have the fibers, the main thing that is done is okay. As I said, the number of fibers, the bundle volume, etc., is difficult to study because the tractography is very noisy, etc. So we don't know if we can find a relation between the number of fiber and some I don't know, clinical score, for example. No, but what has been found to give useful information is the diffusion measures that are calculated from the diffusion tensor. So again, I show you again because I think it's important just to understand that. So once you have the diffusion images, you can calculate the diffusion tensor. This is a model. You cannot see that directly. What we can see is a representation of that like an ellipsoid. But once you have the DTI, you can calculate the fractional anisotropy that is an image. It's an image. And there are also other measures, the mean diffusivity, the axial diffusivity, and the radial diffusivity. Okay. All these measures are images. And also from the DTI, we can calculate the tractography. So you have both information. Once you have segmented the bundles here that I show here, sorry. Okay, once you have a bundle, for example, this bundle, we can extract the FA information from the bundle. Okay, so we extract the diffusion information that is that was also calculated for older brain. Here, for example, is the FA for older brain, but you just extract the information inside the bundle, okay, and you can calculate the mean FA or the mean diffusivity, etc. And these indices are informative about the this person, this patient, or the disease, etc. Because in several diseases, these measures are different from the healthy control. Okay, so that's the way that normally this information is used. For example, here is in bipolar disorder. Once general fractional isotropy is a more advanced model than fractional isotropy, but it's the same idea. You calculate an image, you extract the bundles, you calculate the mean FA, the mean MD, and all the bundles, and you can correlate this information, for example, here with some clinical score. Here, for example, was correlated with psychosis in these patients. And what was found that some bundles had a correlation, a lower FA correlated with psychosis, mainly in the corpus callosum. Okay, and so this is only an example of the studies that can be performed and how the information can be associated. Okay, so we also started to study the short association bundles. That is more new. You can see from 2017. Following the same idea, we try to create atlas of short white matter. This is not new in the sense of the algorithms because of the same algorithms, but what was new that we had to improve the algorithms to extract these bundles because by default these were discarded by all the pre-processing. So that was the work to try to identify this bundle, be more sensitive to the difference in the bundles, etc. And we created this atlas, for example, and I have here a video also. Okay, so I don't know if it's working the video. Okay, here you can see. So these are short association bundles. Then these are not the names are related to the regions that they connect, and it can be used also to perform the same kind of study that I show you. Here you can see the segmentation of some subject. It's not perfect. This data is not excellent, but it's what you can get from a standard scanner. And from these bundles, you can also, I think the main thing you can do is to extract the mean FA and compare. And some studies have been performed on, following this idea. Also, for example, for autism, this is one study that no, this image is from the other study, but in autism we found difference in fractional anisotropy in 13 fascicles associated with social deficits. So that means that in some bundles we found a lower fractional isotropy in autism. And also other difference, for example, were found in bipolar disorder and schizophrenia, and also similarities. Some bundles were show the same behavior and another not, and that was also interesting to study. Okay, and here I have, okay, so these atlas were constructed using deterministic tractography, and we perform a jump to probabilistic tractography. That is an algorithm that try to not only reconstruct the main fiber. I'll show you again. Okay, 1 minute, please. Okay, so here is an example of deterministic cryptography where you put a seed, one or more seeds in any voxel of in all the boxes of the white matter, but you reconstruct the main pathway. You follow the main diffusion direction. So this is deterministic. Okay, but probabilistic cryptography will follow different paths, not only the main, but also some paths that are less probable, but you will have a better coverage of the tractography of the fibers. Okay, because if you only follow the main pathway, you lose some information. So the difficulty with probabilistic cryptography is that the datasets are bigger. For example, you start with 3 millions, but you can have 10 million. So that's why not too much works have performed this kind of study, but we try to do it. It was very long just to have an insight of the how much time it took. For example, only to process the tractography, it took 2 or 3 months. And to apply the algorithms, just to apply the algorithms to calculation, it took 5 months, about 5 months. But what we what we are getting, I think is very interesting. I'll show you that here. So we also found bundles, but these are the bundles connecting the precentral and postcentral gyri. But we have a better coverage of the bundles. You can see the EU fibers like as I don't know. You can find the description in the books that you have a complete cover of the region below the precentral sulcus. So this is interesting because with deterministic tractography, you don't find that. You have gaps between the clusters. So that's interesting, and we are working on that. And also another kind of study that we can perform with diffusion tractography is to perform parcellations of the cortical surface. I show here just one approach, but there are also several approaches. The approach here was to use the bundles, the white matter atlas, segment the bundles in each subject, and reconstruct the regions connected by each bundle. With that, what we got was a high number of parcels that are the regions connected by the bundles, but these are very overlapped. So this was an informatic problem to define the final parcellation, to fuse the bundles, etc., just to find a good parcellation that represent the main regions connected between them by the fibers. And we found that, and what interesting to find, for example, that we could recover the Broca's area and the Wernicke's areas that are connected by the arcuate fasciculus. So is another kind of thing that we can study. And from why it's interesting to create a cortical surface parcellation is because from them you can perform another kind of study that is the connectome, what I told you. And what's the connectome? Okay, once you have the tractography and you have a parcellation, like the one I show here, for example, this one of the cortex. So you can, the parcels are the nodes and the fibers are the edge of the connectome, and you can construct a matrix of connectivity and a graph of the brain connectivity, representing the connectivity of the brain. And from this graph, there are a lot of studies that are trying to identify the brain connectivity from this kind of analysis that is different from just to identify some bundles. So once you have the brain represented by a graph, you can use other techniques that are used to extract information from a graph, like for example clustering coefficient, etc., that try to summarize the structure of the graph. What is interesting is that with this kind of analysis, you can classify different population of subjects also. That is interesting, as and is what I'll show you here. Just an example is not from me, from a collaborator. What they did is they took tractography and they construct the connectomes, or the connectivity matrix, and from the connectivity they extract different graph features like that. For example, what represent the features? For example, uh, how connected are the nodes, if the nodes are highly connected or not, etc. This kind of information. And with this information, you can classify, for example, Alzheimer disease patients. And using different method of classification methods, what you enter to this kind of algorithm is the are not the images, but the features that are extracted from the images. Here, the features are the graph measures. So it's a very indirect kind of analysis. You see, you have the tractography, the connectivity matrix, the graph, and from the graph, several network measures, and from that you can classify and identify if a person, if the data from the person is from an Alzheimer disease patient or from a healthy control. Okay, the strategy here was to use several classifiers and select the most voted option. For example, this person is a patient or a control. And what they find, just here is a summary. The idea is they found an 80% of accuracy when they try to classify Alzheimer's disease and control patients. That means that 80% of the time they find when the images and the features come from an Alzheimer's disease patient or from a control. And the accuracy also is good between Alzheimer's disease and mild cognitive impairment. That is interesting because it's difficult. It's more difficult to differentiate Alzheimer's disease from mild cognitive impairment. And just the last few slides, in a classifier, you can also add other information. For example, here we can apply voxel-based analysis, but on the fractional anisotropy. It means that you compare each voxel between all you take. Or what you do is exactly is we take all the voxel of a subset as a feature vector, it, and for all the subjects. But you will find difference between the subjects that have Alzheimer's disease and the control, and the classifier is able to identify the voxels that are very informative about the discrimination between the two populations. For that, of course, you need to all the pre-processing to perform the voxel-based analysis. Also, you could use ROI analysis to extract mean FA or mean MD, etc. And this is the last example, another work that took 3 levels of information, and that this is another example of machine learning approach. That's why I took this example, because is why I think several work are at this moment being carried out, is using several kind of information. For example, voxel level information that can be the fractional anisotropy, but also the T1, for example, image. And also you can extract information from the connectivity matrix directly from the connectome. That is, I'll show you again, just using the connectivity matrix, and a third level using, for example, the network features extracted from the connectivity matrix. All that you can apply machine learning approach to classify. And this, for example, this work was more accurate than the preceding work because it included more information, not only the network metrics, but also other information from the brain. So this kind of classification can be performed, and also the regions. What is interesting is that the regions that are more relevant, or the features in general that are more relevant to discriminate, can be identified and allow sometimes a better understanding of the disease also. Not all the time, but sometimes you can use this information to understand also the disease, or what you are studying. Okay, that was what I had to show you. Of course, the two works that I show here are not from my lab, but the other works, yes. And this was a work from a lot of people, some lot of collaborators, mainly in France and in Chile also, from students. And that's all that I had to show you. Thank you very much for your attention, and I want to answer any question that you can have. Thank you.